Hi, my name is Rühle. I'm currently a master's student at the DKFC in Germany, Heidelberg, and I'm there with the Stiegler group at the moment. Associating the genotype to a certain phenotype has linked many genetic variants to complex traits or human diseases. But not only can we associate genetic variants to the phenotype, but also molecular intermediates such as gene expression. Further, this gives us the opportunity to interpret these genetic effects. And although many links have been identified, little is known about the impact of genetic heterogeneity on early developmental processes. And since the recently introduced concept of RNA velocity helps us to better understand how development progresses, we asked whether we can extend genotype-phenotype association maps of early development by the use of RNA velocity. To investigate this question, we made use of induced pluripotent stem cell lines from over 100 donors. And first of all, these stem cell lines were genotyped. And then second, we used pooled experimental design to differentiate iPS cells into definitive endoderm. Then the cells were sequenced using single cell RNA sequencing at day 0, so before induction, and then at day 1, day 2, and day 3 after differentiation was induced. Here you can see the principal component analysis of the expression space of the cells colored according to the time point of collection, starting from day 0, day 1, day 2, day 3. Then afterwards, we draw a principal curve through all of these cells and map the cells back onto this principal curve. And this ordering then is what I call a pseudo-time from now on. Then the last step was to assign a cell type to all of the cells. And we did that using the time point of collection and also the pseudo-time that we assigned before. After we've quantified the differentiation progress, we asked whether genetic variants impact this progression. And for this, we ran a genome-wide association study. And interestingly, we find some significant associations between the pseudo-time at day 2 and particular SNPs. And one of these SNPs you can see here on the right. And for example, cells here in this example, cells that carry this TT variant were assigned a higher pseudo-time at day 2 than the other cells that carry the TC or the CC variant. Now we're looking at the dynamics of gene expression and RNA velocity. So first of all, here on the left, you see transcriptional dynamics. Transcription occurs with a certain rate alpha, producing unspliced mRNA. Then this unspliced mRNA is spliced with a certain rate beta, yielding mature mRNA or spliced mRNA. And in the end, this is degraded or translated into protein. So this can be put into a differential equation, allowing us to quantify the changing rate of gene expression, what is called RNA velocity. And to further understand this, we can look at this phase portrait, which is the solution of the differential equation. So for example, when a gene is just upregulated, there's more unspliced mRNA than spliced with respect to the steady state ratio, which is depicted in gray. And then this gene follows the red trajectory. Next, we see that the velocities resemble the expected differentiation progress, and also that the highest velocities are observed in mesendoderm and pre-endoderm state. Looking at a single gene, GATA6 in this case, we can also see that expression and unspliced mRNA abundances follow different dynamics. And from this, RNA velocity is then calculated, and we can either calculate it using a dynamical model or the deterministic model. But in both cases, we see that RNA velocity goes up before expression does. lastly, we associated the obtained molecular intermediates from step 3, expression, unspliced abundances, and velocity, with genetic variants in a cell type specific manner. And for this, we used a linear mixed model. Looking at expression QTL, we find per cell type over 1,000 genes that have at least one QTL, and these 1,000 are depicted in green here. But the number of genes with a QTL is lower when we map unspliced transcript abundances, or which are red, or RNA velocity, purple. But interestingly, when we intersect all of these genes identified here, we find that the overlap of these genes is smaller than expected. And we identified genes that have at least one velocity QTL, but not an eQTL or an unspliced eQTL. Moreover, we tested the stage specificity of QTL, and whereas we find similar patterns for eQTL and unspliced eQTL, the relative number of QTLs that are specific to only one stage, they're these light purple, decreases when we look at velocity QTLs. So the conclusion is that cis-QTL mapping of RNA velocity and also unspliced transcript abundances do extend genotype-phenotype association maps. So thanks for listening, and feel free to reach out if there's anything I can help you with.